Thank you, Fernando. Hello, everyone. So my name is Neriah Nicholas. Today I'm going to be talking about a sub-project that's a part of my master's thesis, which is on the assessment of morphological differences and genetic diversity in Florida Pompano. So first, while we are all here today in this room, we are here to discuss the warm water marine finfish aquaculture, which involves the breeding, rearing, and harvesting of finfish in order to either repopulate a species and or to harvest them as a food source. So, as you can see here, over 50% of our seafood comes from America, comes from aquaculture, but in America it only produces less than 1% of aquaculture for the world share. But there is a gradual increase in aquaculture globally, and the demand here is high and the importation of seafood is high. So the demand to increase the industry is very high, and our funding source, the USDA, their main goal is to actually support the expansion of the industry. So why Florida Pompano? Well, they are a model species for warm water marine finfish because they are a popular catch with fishermen. This is actually a local fisherman off the west coast of Florida who won a contest with a Florida Pompano. And they also have a high market value. On estimate, they are normally about $22 to $26 per kilogram, but in some locations they can range higher than that. They are found off the east coast of the United States from Massachusetts to Florida, around the Gulf of Mexico, down Central and South America. The juveniles are normally inhabiting the surf zone, and then they travel offshore and when they reach sexual maturity, and then they come back inland to spawn where the young float freely in the water column. They are model species for aquaculture because they can tolerate a wide range of salinities. They also can withstand dissolved oxygen levels that are low and temperatures that are high. They have rapid growth rates, and in our systems, they reach market size at around 450 grams is when we're able to sell them. So now shifting gears a little bit from that background, we're going to go into genetics. So genetics is widely applied throughout aquaculture. I'm specifically focused on genetic selection using genetic markers for key factors such as growth and sexual maturity for aquaculture farmers. And so this is actually previous research where they took brood fish and they tracked the spawn to see the length of them, and then they genetically selected certain brood fish and tracked the spawn to see the length of them. And as you can see, the genetically selected was a higher length on average. So the specific genetic markers I am looking at are microsatellites for my first one, which are tandem repeats between 1 to 4 base pairs. So here you see 4 base pairs that are repeated. Here's a previous published study tracking genetic diversity with microsatellites where they were tracking different Trachinotus species, and the different colors represents the genetic variation or the genetic overlap between the species. The next genetic marker I was looking at were single nucleotide polymorphisms. I'll refer to them as SNPs from now on. They are variation in DNA for a single base pair change. So like here in the first sample, there's a G, and then in the second 2 samples, it was replaced, but only one was replaced. And here is a principal component analysis, which is a type of scatter plot commonly used for SNPs to visually see the overlap in genetic variation. We were looking at 2 populations, Gulf Coast and East Coast Pompano, because scientists believed that they were genetically distinct from one another. But as you can see by the amount of overlap from the blue represented the Gulf and the black, which represents the East, there is no statistical difference between these 2 populations based off of our sampling of the wild-caught Pompano. So my overall objectives were to first characterize the features of the captive-bred Florida Pompano, and then compare the differences in the growth of juveniles from a single spawn, and then to try to help develop techniques to evaluate the relationship between genetic diversity and growth and size. For this study, this was opportunistic sampling. While I was doing my routine sampling for my master's thesis, we found in a single cohort that there were fish that had concaved head sizes. So I took 10 of the concaved head size fish and 10 of the normal fish and measured 11 morphological measurements. The total length, the standard length, the depth length of the fish, the head size represented here, the operculum length, or like the gill slit cover, from the top of the fish to the eye, from the bottom of the eye to the bottom of the fish, the eye diameter, are some of the measurements I took. Here are some of the samples from this study. The first 6, or the red circles, represent the concave morphology, as you can see here. It kind of looks like you just took your thumb and pushed it in. We did not do this though, they were like that. And then the green circles are the last 6 fish. These are more of a slope. That is normally what you see in juvenile pompano. For reference, if you can't tell based off the picture, the juvenile pompano were about 80 to 100 millimeters in length, which are about the palm size of your hand, or maybe yours. I have tiny hands. And then after we measured all of them, we took pectoral fin clippings, which if they were live, it would not harm the fish, but these were cold in order to take all of the measurements just for ease. Then I did the DNA extraction in-house by following the Kaijin protocol. Here in the lab, because I had froze the samples after collecting the pectoral fin clips and waited to do genetic analysis, I re-clipped them so I could possibly rerun the DNA extractions if needed. And then I ran the sampling, but you can do DNA extractions right away. After the extractions, I test for the quality and purity of the DNA, and then I shipped it out of house for rad sequencing. I do not have their exact protocol, but this is like an overview of what rad sequencing is. So it's restriction-associated DNA sequencing. So first you take the DNA and you digest it or fragment the DNA. Then adapters ligate or bind to the fragments of DNA. Sometimes there is additional adapter slicing. And then you take the fragment sizes and you select for them. They are amplified repeatedly and then sequenced, and then you get your raw rad-seq data. So shifting gears into some preliminary results, even though you saw that concaved indentation visually, there is no statistical difference between the concaved morphology and the normal morphology. Additionally, within each of the morphology types, yes, there's a statistical difference between the individual if you were to compare them. But obviously, the length of the eye diameter is going to be different from the length of the total of the fish. We are looking at the actual morphology types as a whole, which were not significantly different, represented by the p-value, and if you just look at the average mean. So my next steps for this is to actually assess the genetic diversity by taking those raw reads from the rad-seq data, doing quality control checks, then trimming any of the adapters, the barcodes, the pore base pairs, the pore sequence reads that we got from them, and then doing a genome alignment. We actually have a published genome assembly with NCBI already, so we'll be using that as our reference genome. And then if that doesn't necessarily align well, there is other reference genomes that we can use, but this is the main one we're going to look at for now. And then we're going to use genotype samples. So my future directions for this project is to first look at the genetic markers. Are they associated with morphology? And if they are, is there one that's more advantageous for the fish? The way we would look at this is we would reassess if the concave morphology yield has a higher or lower dress out percentage, and for the fillet yields as well, because that's the whole point of fish farmers. They want the bigger, the best fish. And then additionally, if this concave morphology we come across again, instead of culling them and harvesting them as juveniles, I would like to try to, yes, it would take longer, but I would like to try to do the morphological measurements while they're still alive and track them throughout grow out and pit tag them, so I can see, does that concave morphology now have significant difference as they get bigger, or does it never have any significant difference and it just changes the dress out yield, or maybe it doesn't change at all and it has nothing to do with the fillet. So in summary, although there is a visual difference, there was no significant difference between the head types. There was no significant difference between the morphology types. The individuals were not significantly different from each other, and I'm still in the process for the genetic diversity steps. I would like to give a huge thank you to the USDA for funding this research. We would not be able to do any of this research without them. I would also like to give a huge thank you to Aquaculture even if I had funding, I wouldn't have the fish without them. And if you scan this QR code, it takes you to their main website where you can either look at them or you can actually get recipes from them for Florida Pompano. And I'd like to thank my institution, Florida Atlantic University, for having me as a master's student, and the Harbor Branch Aquaculture Department for assisting me with this project. Thank you.
Time for questions?
Yes.
Did you say you caught these guys in the wild?
No, these were all, oh, she asked if we got these from the wild. This specific study, the one that I sampled from, was sampled from Aquaculture, the fish farm, so they were raised in-house. But the previous studies in the background information, those all were studies from Wild Pompano.
Did you find any literature regarding the concave head? Like, has that been documented?
She asked if there's been previous literature published about the concave morphology within the Florida Pompano. No, there is concave morphology within other fish, which is why I wanted to look at it, because it is crucial in certain species. So like trout, for example, there are studies that is done with it, and it does actually decreased or concaved head increases the fillet. So that's why we wanted to look to see if this is significantly different. Because it wasn't significantly different, we didn't look into it further. I don't know about that. Thank you. Any other questions? Yes, Maggie.
You didn't find the answer in the genes. Do you think it's invisible to a lot of things?
She asked if I don't find a difference in the genes, do I think it's anything environmental in the tanks? The answer is yes to that, because at Aquaco, this morphology was only observed in one of the cohorts and only within 2 specific tanks, and there was 4 tanks for this cohort. So we think it possibly could be. The only problem is it's never happened there before, so we're just looking into it just to see. Go ahead.
Have you asked Chris for our journalist? Do we have the issue?
She asked if I've asked Chris, which is at Harbor Branch in our aquaculture department, if we've had that issue. I have not asked him, but I've asked Paul, and Paul's never seen it. So it was very interesting to see. There are different like head morphology types that are found within Florida Pompano that mess up like the skeletal system that sometimes messes up the head, but never like this concaved indentation. Yes.
Follow up on that. We took some of those same things and raised them in my hatchery just to see what it was from.
Mhm.
For Joe.
Mhm.
Didn't have any of that. So it's not.
Okay. Okay. Thank you.
Any other questions? We have 2 more minutes. Okay, good job. Thank you.
Thank you, guys.